Synthetic biology is an emerging field of science defined from two directions, from the top down and from the bottom up. In the first case, researchers tried to create new function and new application, reprogramming existing organisms such as bacteria and yeast. In the approach from the bottom up, we tried to synthesize from scratch living organisms with some or all of the characteristics of living systems. Research in synthetic biology requires the skill and expertise of many different disciplines, using the tools from molecular biology, chemistry, physics, microbiology, and even engineering. The objectives of synthetic biology are diverse, but overall we hope to understand the fundamental properties of all living things on Earth. From the perspective of synthetic biology, we study how living organisms are organized and function. We do this by making simplified models to understand how living systems are organized as far as their complexity, their organization, and their evolution. Today we have several models of artificial life constructed in the laboratory. Each has some fundamental properties of life, metabolism, genetic information, or membrane, that resembles living systems. Nevertheless, these models are very far away from having all of the characteristics of living systems. In the future, we may have several forms of technology created by humans that may be considered living. For our synthetic biology experiments, it is believed that the risks are low because our experimental models contain only some of the characteristics of life and are not yet alive. For example, the experiments that we do today in the laboratory do not contain DNA, and therefore there is no risk of out-of-control evolution. At the same time, it is important to communicate all of the benefits of this type of technology.